This is a groundbreaking effort to compile an atlas, a full collection, of vaccine candidates for Group A Streptococcus, by a systematic comparison of a large number of genomes. Group A Streptococcus, GAS, causes a very high global burden of disease. Despite over 100 years of research, no commercial vaccine has been developed yet. This study identified 299 GAS genomic phylogroups across 22 countries and found that 13 antigens are likely to be effective global vaccines. Let's come to the details. This research investigated the largest and most diverse GAS database to date, 2019, consisting of 2,083 isolates. The EMM gene encodes the major surface M protein, a virulent factor of GAS. The database showed 150 EMM types, 347 subtypes, 39 M protein clusters, and 484 multi-locus sequence typings, MLSTs. The coding sequences that cover over 99% of isolates are called the core genes. The database showed that the GAS genome has 1,306 core genes. Among these core genes, 416 core genes don't have recombinatorial evolutionary histories. By aligning these non-recombinogenic core genes, we constructed a phylogenetic tree for all genomes, F1A. Using population network analysis, we identified 299 distinct genetic clusters, phylogroups, of which 206 were represented by 2 or more isolates. Plotting the pairwise genetic distance, we found that many EMM types, EMM subtypes, and M protein clusters do not share close evolutionary history. This indicates that these markers alone are not enough to classify global GAS populations. By evaluating homologous recombination within each phylogroup, we observed that each phylogroup has around 5,500 SNPs, single nucleotide polymorphism, and small homologous recombination is the main driver for evolution. Whether any gene marker is related to specific clinical symptoms? To answer, we analyzed the correlation between lineages and clinical features. The data showed that the disease tendency is not limited to a single lineage, SF5. The study also identified 5 virulent factors associated with invasiveness. However, they are needed to verify in clinical studies. Next, we evaluated 28 known antigens for the capability to become a global GAS vaccine. By screening homologous sequences across 2,083 genomes, 13 antigenic genes were found in over 99% of isolates and showed very low sequence variation, less than 2%. Therefore, these antigens are strong candidates for global GAS vaccines. Among these antigens, the M protein is the highest variant, which in turn confirms the challenges to target M protein as a vaccine candidate. This study also investigated the polymorphism of antigenic structure within the global GAS population by examining 2 well-known antigens, the streptolysin O and C5a peptidase. The variation of amino acids in streptolysin O is lower than in C5a peptidase. This is confirmed by the number of hotspots in C5a peptidase, which is 4 times higher than in streptolysin O. These data indicate that some regions in these proteins are evolving adaptively. To sum up, this study developed a groundbreaking platform for analyzing candidate antigens. It revealed that recombination and selection pressure are extensive across the GAS population and identified 13 vaccine antigens that can be a global vaccine. Finally, this platform for vaccine design is equally applicable to all known GAS antigens, as well as to the future ones.